So today we're going to talk about feedback mechanisms and how organisms use them to maintain dynamic homeostasis. Homeostasis refers to maintaining the internal environment of an organism within parameters that sustain life. This can mean things like stabilizing blood pH or blood sugar. For today's example, we're going to look at how these concepts are relevant in human energy metabolism. We're going to start out by looking at the core pathways involved in the utilization of our two primary energy sources, which are glucose and fatty acids. Starting with glucose, this first step in using glucose is to break it down into 2 3-carbon sugars. This split produces a little bit of energy, but not very much. While this first step takes place in the cytoplasm of the cell, so the main body of the cell, this next step takes place in the energy metabolism specialist organelle in the cell, the mitochondria. So we're going to draw a line between what we have so far and the rest of the pathway to remind us that we're in 2 different places in the cell. So now we've got that. We transferred that 3-carbon molecule into the mitochondria, where it gets transformed into a 2-carbon molecule. This step also produces a tiny bit of energy. The carbon that is lost in going from 3 to 2 carbons is released as CO2. This 2-carbon molecule that we now have, it's known as acetyl-CoA, and it is the entry point for the citric acid cycle. For our purposes today, you can effectively think of the citric acid cycle as a machine that takes in that 2-carbon molecule and extracts the energy from it. In order to do that, it does take a 4-carbon molecule, combine it with the 2-carbon molecule, producing a 6-carbon molecule, which then becomes a 5-carbon molecule, and back to the 4-carbon molecule. That loop makes it a cycle. But the real key thing you need to know here is that this process releases those 2 carbons as CO2 and harvests a ton of energy in the process. So that's glucose. So let's look at fatty acids. Fatty acids don't need any work done before they get transferred into the mitochondria. They come into mitochondria as is. Once they're in the mitochondria, fatty acids are essentially just a long chain of carbons. What we do with that long chain is break it up into a bunch of 2-carbon pieces. In fact, the same 2-carbon pieces that we had from breaking down glucose. These 2 carbons can then be fed into the same citric acid cycle. So from what we have so far, it doesn't seem like using glucose or fatty acids for energy should be all that different. In the end, they both feed into the citric acid cycle, so it's all the same, right? However, there's a few things that make glucose and fatty acids very different in terms of how our body uses them. In particular, it matters that not all cell types can use fatty acids directly. First of all, red blood cells are special and don't have mitochondria. That cuts off everything below our magic line, which means they're left with basically just this first step. Secondly, our brains, which are very energy-hungry organs, aren't very good at this step where they turn fatty acids into the 2-carbon pieces. They do have a citric acid cycle, they have mitochondria, but taking fatty acids into the cell and turning them into something that can be fed into the citric acid cycle, that they don't really do. Additionally, I should note that while glucose is good for quick use, because all the cell types can use it, fatty acids are way better for long-term storage than glucose. There's a storage form of glucose that we'll talk about in a minute, but fatty acids store energy in a much more compact way. So we prefer to use fatty acids as kind of our backup source, and glucose as our default. In fact, this is basically how the regulation of energy metabolism works. We have a mode where we're using glucose for just about everything, and a mode where we're using fatty acids for as much as we can, and glucose only for the essentials. Now we're going to look at the goals for the regulation of each of these 2 different modes. When the whole body's running on glucose, we want to have a lot of glucose in the blood, but we also are concerned about too high of blood sugar, because blood sugar that is extremely high is dangerous for the body. It's the job of insulin, which runs the show in glucose use mode, to control blood sugar levels. So now let's talk about fat. We know fat can't be used by all the cells in the body, so you can probably guess what we need to do in fat use mode, and that is to ensure that we supply our picky cell types with something they can use. So the hormone in charge of this mode is glucagon, which ensures the feeding of our picky cells. So now let's look back at our pathways and see what happens when insulin or glucagon is running the show. First, we have insulin. All of the cell types that are capable of running on fatty acids run on glucose only when insulin tells them, hey, we're in insulin mode, there's tons of glucose available. So this step where you take up glucose from the bloodstream, that is insulin dependent in those cells. So we're going to draw glucose here and show it coming into the cell. Once glucose is in the cell, it's processed through glycolysis, breaking it up so it can go into the citric acid cycle, is also directly promoted by insulin. A lot of the other effects of insulin happen specifically in the liver, so we're going to change colors so we can keep track of that. The liver wants to ensure that glucose levels don't get too high and prepare for times when glucose isn't plentiful. So the liver will direct the storage of glucose as its short-term storage form, glycogen. We only store enough glycogen for a short period of time, a few hours of intense activity or a bit longer if we're not doing anything super intense. But this storage source is very important. Insulin signaling also tells the liver that it's okay to take materials out of the citric acid cycle to form new fatty acids. Fatty acids can then be sent to the fat cells and stored as fat. It wouldn't make sense to also be allowing fat to be broken down into free fatty acids at the same time that we're building new fatty acids. So insulin directly blocks this process in the fat cells. Now let's hide the insulin pathways and look at what happens with glucagon. Glucagon is telling the body that it's short on glucose. So one of the first things it's responsible for is promoting the breakdown of fat into fatty acids. These fatty acids will be put into the blood so that tissues that can use fatty acids for energy have them available. These next steps apply to the liver, so we'll switch colors slightly again. The liver is concerned about making sure there is enough glucose available for the tissues that require or prefer glucose. So it does this in two ways. First of all, it can break down its stores of glycogen to produce glucose, and it can also pull materials out of the citric acid cycle and make new glucose. Both of these sources then can be released into the bloodstream. These sources for glucose production, though, are somewhat limited, and we have so much energy in the fatty acids. So we actually have one more thing the liver can do to help out the brain. Remember, the brain has mitochondria and can use the citric acid cycle. It just can't chop up fatty acids into little pieces. So the liver produces from fatty acids small compounds called ketone bodies that can feed into the citric acid cycle. These are released into the blood so that they can get to the power-hungry brain. So now if we look at everything together, you can see that insulin and glucagon are basically opposites. And maybe you can begin to see how this all works in the body. The release of insulin is triggered directly by high blood sugar. So when we have insulin, we have glucose being pulled out of the bloodstream by every cell, and we have glucose being stored as fatty acids and glycogen. This will allow the blood sugar to come down. When blood sugar comes down enough, insulin stops being released. When insulin's not being released, then glucagon is released. Glucagon's release is directly blocked by insulin, which makes sense. We can also see how the whole system would break down if we don't have insulin. Without insulin, we don't have a lot of cells pulling glucose out of the blood, only the ones that aren't insulin dependent. We also aren't storing it as glycogen or burning it up in glycolysis as well. Also, without insulin, we don't make new fatty acids, and we don't have anything to block breaking down fatty acids from fat, even though we don't need the energy. So this leads to this weird state where even though there's plenty of glucose, the body thinks it's starving because insulin isn't blocking glucagon, and glucagon is telling the liver, freak out, make energy, we're starving. And so all those fatty acids get turned into ketone bodies. The parts of the body that are supposed to use ketone bodies, though, don't need them because there's glucose. This is exactly what happens in type 1 diabetes, where a problem with the pancreas means you produce absolutely no insulin. Glucagon directs the body to act like it's starving, producing as much glucose as possible and dumping it into the bloodstream, meanwhile producing massive amounts of ketone bodies. So now you have extremely high blood sugar and also all these ketone bodies which actually are slightly acidic. This lowers the pH of our blood, producing a very dangerous condition called ketoacidosis that without insulin treatment can be fatal. And that's where we'll wrap up for today. We've looked at how insulin and glucagon work together to regulate blood sugar levels and manage our energy metabolism. And we've also looked at what goes wrong when a piece of a feedback regulation system is missing.